entitled A Framework for Intelligent Non-Animal Methods for Safety Assessment, and it's got about 7 different ways of referring to it, and I don't think anybody in the committee really knows what it's officially called. So I'll go through how did we end up with a project on this topic, why do we feel that it's timely, and what are we doing to try to take it forward. And so, as so many topics in emerging toxicology, we would argue that one of the seminal points was the publication in 2007 of the National Research Council's report on toxicity testing the 21st century, a vision and a strategy, followed up in 2009 by Science and Decisions. But I would suggest that whilst the 2007 report certainly crystallized a lot of thinking, it also represented a convergence of thought. And even without that report, it's likely that toxicology would have moved in the direction it's moving in. And that direction was a very large scale, perhaps not global, but certainly widely dispersed across geography, a number of initiatives to generate non-animal methods, which is code for saying in vitro and in silico, to replace animal methods. But this was fundamentally different from the initiatives that had gone before in the so-called 3 Rs, reduction, refinement, replacement, which in the US is coordinated by IGVAM and in Europe is coordinated by IGVAM, which is a very formal process of one-to-one replacement. This was something quite different. But the problem was that you had a whole group of individuals in the public and the private sectors generating lots and lots of new assays or methods, but this was largely uncoordinated. In the last couple of years, we've begun to see some sort of interaction and coordination, but certainly in the early part, in say the 2008, 2009, we were seeing a lot of assays being generated in laboratories in the US and other groups in Japan or in Europe generating their assays, and they were overlapping and different, and there was no real coordination. So in 2014, HESI came up with a proposal to try to identify a consensus across the initiatives and provide a scientific confidence in the methodology. Now, I need to make a very important point here. This initiative was to develop a framework to establish the fitness for purpose, largely for regulatory decision-making of emerging methods. It was not to develop a set of methods to replace animal testing. That's what's ongoing and is somebody else's activity, but this was not part of the HESI initiative here. This was to say, if you're going to argue that you now have a new method that adds value and can replace some of the animal data in regulatory decision-making, what sort of information do you have to provide to convince me as a regulator or decision maker that is fit for purpose for that objective? So that's what the framework was about. So why now? Well, there was a need to determine a consensus across all these initiatives, and this was canvassed across the various organizations that are involved in this type of work, such as the US EPA, the UK NC3Rs, the European IGVAM and JRC, and there was a general agreement that this initiative would actually add value, that there was a need to bring people together to develop a framework of this type, because there wasn't a concerted effort, even at OECD at the present, to do this. It would therefore provide scientific confidence in the methods that were emerging, and that it would guide the future development of those methods by laying down the principles that would be required to establish their fitness for purpose. So how is HESI addressing this issue? We started with one proposal, and we intend to come back to one framework, but in line with the rule of threes, we set up 3 subgroups. One was on one was on performance characterization, one was on model predictive performance, and one was utilization. And it took us a while to define the scope of each of these groups, but I think one of the simplest ways of explaining it is to say that if your adverse outcome pathway activity at the present time. You have a series of non-animal methods to evaluate the individual key events, including the molecular initiating event. And so performance characterization would be establishing that that method is adequately defining that key event. Model predictive performance is showing that when I measure my different key events, I'm able to reconstruct an AOP from that information. And utilization is then demonstrating that that AOP can be used in a meaningful way for the particular problem that I'm faced with in risk decision-making in risk assessment. And we recognize that there's a spectrum of different issues that have to be addressed, from simple screening or prioritization of chemicals through to safety assessment or even quantitative risk assessment. And that will require different criteria depending on the issue that's being faced. We're pleased that there was a substantial interest amongst members and stakeholders, and over 60 different people are involved in the various sub-teams, and they participate regularly on our monthly conference calls. We put together a steering team. The project is co-led by myself and Craig Rowlands. Craig, however, has recently left the Dow company to take up a new position, and because the organisation he's working with is currently not a HESI member, although he hopes that he can do something about that, we have looked to identify another co-chair, and Doug Wolf from Syngenta has kindly agreed to do that. And the 3 sub-teams are co-led by industry and academic, they're co-led by 2 individuals, and each of those 6 individuals are on the steering team, together with a number of other representatives from various organizations. And you can see there's a good mix of public and private, and including a number of important regulatory government organizations. And we're very fortunate that we actually have somebody from the European Federal Agency for Medicines and Health Products, as well as from the NC3Rs in the UK. So how are we doing this? Well, we decided what we needed was a set of criteria against which we would judge these 3 different aspects of method characterization, and that we would do this by looking at what's been done before and creating a matrix of the criteria and the different applications of the methods, for example, starting with something as simple as simply prioritization or screening and running up through to the more data-intense or more demanding applications such as safety assessment, and try to fill that matrix in to the extent possible. So determining the criteria, each of the 3 subgroups came up with their own set of objectives and questions. These were substantial overlap, it has to be said, but the interpretation of the criteria would be different by the different subgroups. Determining the set of sub-criteria, we extracted information from the literature, we applied our own considerations and thoughts to what would be appropriate and relevant, and then determined a consensus on the minimum criteria required. And it has to be said that this is a work in progress, so we have a list of criteria, but we're going to stress test these with case studies, and I'll discuss that in a moment. So this is the matrix, and this is simply the shape of the matrix. It's not specific. So for example, the criteria we have would include issues such as what endpoints being measured, how is the biological verification of fitness for purpose, what is the minimum applicability, and that would be filled in across for different assays for different types of application. And here are 3 examples: the estrogen receptor binding assay, which is a very, I hesitate to say simple, but it's a very semi-qualitative assay. Then the HERG assay, which is cardiotoxicity, which is more quantitative, and then in vitro to in vivo extrapolation, which is even more quantitative. These are just examples. And then Having determined these criteria, these were discussed at length on a series of phone conference calls. We have now agreed what they are, and the matrices are all filled in, and we're now choosing case studies, and these are going to be used to stress test the individual components of the framework, these 3 different sub-components, and they're currently under development. We've chosen the chemicals or substances which will be used for the case studies. I would say, certainly in my own group, it's 3 at the present time, and I think it's 3 or 4 for the other 2 groups as well. And the goal is to have case studies that are actually applicable to some extent across all 3 of the subgroups, so that even though they're being developed by the method performance characterization subgroup, it would have some relevance to the other subgroups as well, and vice versa. And then we're going to ask the question, are the criteria met? And if not, what criteria are missing? And also another question is, are there some criteria in there which are actually not very helpful in reaching a conclusion? In our own case, the utilization sub-team, we will go further and we'll ask that given there are very few case studies where there is actually regulatory acceptance of these methods at the present time, but we have examples of where there are a lot of data on the methods, we're asking the question, what more would be needed to get that method to the point of where it would be considered fit for purpose? And then these case studies will form the basis of a workshop which will be held in the fall of this year. And this workshop will be an opportunity to receive feedback from outside organizations. We're going to invite a number of speakers from various organizations who have not been involved in framework construction within HESI to provide their viewpoint. They will be provided with the framework, the draft framework, and the case studies, and they'll be asked to come along with, provide their critical input to their views on the utility or otherwise. What are we missing and what have we got right? There'll be breakout sessions to disseminate the information discussed, and then based on feedback from that workshop, we will then revise and adjust the framework. And then the objective, of course, will be to publish that as a contribution as to how this might be done in the future. So, this working together, and then we'll get at least one publication reflecting this framework for the intelligent non-animal methods in study assessment. So, in summary, we're trying to look across initiatives. We're not. It was agreed from the outset that it's really important we don't reinvent the wheel, that we look to see what else has been done in the area, and we harmonize to the extent possible. Create an umbrella framework which is consistent, transparent, and generally accepted across sectors. Input from multiple industry sectors, agrochemicals, industrial chemicals, pharmaceuticals, etc., and different backgrounds, public and private. It should be acceptable across different communities through peer engagement of the and a judge assessment of the criteria proposed. Provide a future guide to organizations where they are thinking of approaching the replacement of animals with alternatives, and this will then give them a strategy by which they can judge the utility of those methods for the intended purpose. I think that's human. So thank you.
Thank you very much, Alan, for that succinct summary of where you're going, where this is being taken. Questions? Or actually, I'm sure Alan would also welcome discussion, inputs, comments.
They're all sold.
I have one question, Alan. When you come to do the evaluation of the criteria, is that a binary decision, yes or no, the criteria is met, or have you considered, do you intend to score it, or? I was trying to imagine what you would populate your tables with. Would it be a tick or a yes or a no, or?
It will vary, because some of them are quantitative criteria. So things like how many reference chemicals you've got in there, how many true positives, how many true negatives. So that's a quantitative answer, and we wouldn't say tick, you've got 80%, because that will vary.
Yep.
So that information will be captured. But other times it will be, have you given a protocol where some other people can follow the method, yes or no? Yeah.
Okay. And have you thought about how you will then ultimately make a judgment on overall whether those criteria have been fulfilled to an adequate level?
This is we're just at the beginning of the case studies.
Yes.
The impression we're getting is that it will be easier for the first two sub-teams for the performance characterization, because people are doing that. I mean, this is just trying to formalize that. And then the model prediction, there's some work on that. But I think when it comes to utilization, when we looked across all the different examples we had, when you think about it, there's only about two that have been accepted. Sensitization, recently, and there's the recent proposal that the 18 parameter assay parameter ER predictive model that EPA have come up with. The general view is that's seriously overspecified, and so that's something we'll look at as well. That's one of the criteria. What can you leave out? But apart from that, it's going to be, why is it that these assays are not being used at the present time? And again, I stress, these are a means to an end. We're not interested in, advising people on individual assays. It's about criteria by which you judge a new assay. So these are just a means, case studies to get that information.
Sarah?
So since you have ICVAM and ECVAM and JECVAM involved through representation on your program, if you project out a year or 2 years from now, or at least from that workshop, do you see them citing these papers, or do you think this is something that would actually ultimately result in some changes in the kinds of criteria that they actually put forward independently?
I think it's the latter. I think it will inform the criteria. I'm hoping and thinking that they will look and it will provide a solution to a problem they have. If you talk to people from these organizations, they fully recognize that we're never going to get to where we need to be using the process that they've been trying to use, which is the formal validation in a multi-laboratory trial. It's taken us, 30, 40 years to get 4 binary assays for topical exposure. 40 years to get sensitization in skin and eye, and irritation. And so I've spoken to people from ECVAM, and they are very excited about it, and they want us to come and talk to them about it and to take on board the lessons that we've learnt and hopefully inform their process. So I think it will be a contribution. And ICVAM was involved from the beginning.
Thank you very much, Alan.
Sorry. It was
Just you almost escaped. Just to comment on the importance of the timing of this. I think at least the Americans in the room will know most likely that during this Congress, we're almost certainly going to replace the piece of legislation for chemical management, the Toxic Substances Control Act. There's one senator holding it up, and I hear that he's going to release that hold. There's a requirement in the new piece of legislation that says there has to be an emphasis on non-animal testing. And EPA has 18 months from enactment to develop new policies, procedures, and guidelines, including how to test for safety and for risk. So I think this is really going to be timely. They're going to be very interested, I think, in this work. This framework should help to inform which assays will be viewed as credible and which ones maybe are less so.
Thank you.